So I thank the organizers for giving me this opportunity to speak in this virtual symposium. Thanks to Abhishek and others in the team. So my talk today is on extending the horizons of biological function inference, leveraging protein dynamics. So the whole idea of giving this talk is to try to simplify things. We know, we live in an era where we have a lot of data, and coming up with methods that are simple enough to understand things is the need of the hour. Otherwise, we will get overwhelmed with data. So the outline of my talk first is that I will tell you the motivation for whatever I'm doing, the key concepts involved, the implementations and the applications, and then some conclusions. So if you have a protein, and you want to determine its function, so typically what we do, so you may have a sequence at your hand, you may have a 3D structure, or you may have gathered some high throughput data. So if you have a sequence, you may want to do some alignment, and the alignment could be global, local, or you might want to use some genomic context based method to infer functional linkage, or you may want to use some features and then leverage machine learning. And you can get subcellular locations, functional class, and so on, which allow you to understand what's going on with the protein. And similarly, if you have 3D structures, you'll have again global and local, matching, and then you would have a 3D motif, or you can also leverage structural features here to train your algorithm and come to some inference. If you have high throughput data, then gene expression or protein-protein interaction information can allow you to gather new insights into the functionality of the protein. But if you look at whatever we have been doing, there's a catch in it. The conundrum is that the protein function is a biological activity. So what is an activity? It's actually a dynamics in space and time of the protein molecule. And it is governed by the physicochemical properties of the parts in action and the surrounding environment. But given that we have a limitation, we are currently only using static information to infer function. And as in the case of protein, where you're leveraging, primary data or the tertiary data or the quaternary data that's available with us. And we typically use what is called the sequence structure interaction, linkage to understand function. And whenever motif information is available, we try to leverage it as well. So this is currently where we are. And you may be knowing that, that 98% of whatever we understand about functions of proteins is computational. And this is, as per the latest available information in the GEO database, and only 2% is experimental. So understanding function of new proteins, trying to find out more functions of what they do is very important. And I'll show you, why, dynamics is so important. So this is one simple example that is at hand here. So we have a structure for an avian defensin protein. This is from chicken. So the defensin protein, and the NMR structure for the wild type on the left-hand side. And you can see this protein here, it's very flexible, and it has the activity that you'd expect in the wild type. It's available in the PDB database to LG5, and there's only one single, mutation from lysine to alanine, and you can see that the the fluctuation has entirely stopped, and the activity is almost gone. So this tells you, that the flexibility of the protein is very important for function. And the region where this mutation happens, where this alteration happens, determines how critical it is. Now, if we look at only structural perspective, you know, uh, we may not be able to gather simply by looking at the mutation K to A. Most of us typically, when we do mutations, we try to replace any amino acid to alanine, hoping that that mutation will not alter the structure, so that we will know the function. So, so this is not straightforward. There's another example from work that we have published recently in scientific reports this year. This is from a serpin protein. So we have a single mutations, and these mutations are all seen in patients. So the serpin is an antitrypsin protein, which is metastable, but the wild type protein, is not so prone to aggregation. But when you have a single mutation, for example, glycine, sorry, not a glycine, the glutamate to lysine, it becomes pathogenic, which means that this protein is no more, very stable. It aggregates, and you get a disease which is called chronic obstructive pulmonary disease. Recently, we have identified another mutation from glutamate to aspartate, and this mutation is highly prevalent in Kashmiri population. And we have found that this supposedly benign mutation, because you can see there is no change in the functional group, both are acid, but still the protein is prone to aggregation. So these kinds of analysis is very difficult when you have only sequence information, and unless you do dynamics kind of analysis, it's very difficult to understand where the changes are and how important they are. So as you can see in this plot here, I have shown you the root means the dihedral fluctuation of the backbone and the different secondary structures in the 1 microsecond simulation. You can see one change in amino acid, and there are several parts of the protein structure which are different. Most importantly, the electrostatic signature of the molecule also changes. Another interesting aspect that one would like to keep in mind is that there is another class of functions which we typically don't bring to our attention regularly, but are very important, which is called the moonlighting functions. So what are moonlighting functions? Moonlighting functions are functions that are done by the protein other than its main function that it has inherited from its ancestors, which means the orthologous function. For example, you have E. coli put a protein, it does a different function when it is bound to the cell membrane as opposed to a DNA. There's another example. The enzymatic activity is different when it's inside the cell, but this converts to a growth factor when it is secreted. The protein in C has a different function when it is in an endothelial cell as opposed to a neuron. Similarly, if you have a different substrate bound, proteins can switch activity. If the multimer or a monomer in different oligomeric states, it can have different function. Similarly, different complex formations and different binding sites. So the number D, E, F, and G are more commonly known, but obviously A, B, C, D are not so common. But the emerging view is that the proteins are versatile, so much so that when they change their location, the environment change, they can, you know, recruit different functions that is present inherently in the sequences that from which they are coded. So the quest for understanding protein function from protein structure is not new, right? Many have tried to do this, and one of the papers published about 8 years back tried to do this in a comprehensive manner, where they try to find out what kind of analysis would be suitable, trying to link protein structure and function. So this is one of the most comprehensive attempt to link the dynamics to function, and where they tried to decipher 34 features, which means there's a 34-dimension vector to match the dynamics of two proteins. So typically these dimension vectors characterize the eigenvalue spectrum of the protein, analyze the principal component of the trajectory, the ruggedness of the free energy landscape, and the atomic fluctuations. So these attempts have been ongoing. So we also started our project, more than a decade back. My graduate student who joined in 2009, I gave her this problem, and we decided that we'll pursue such a project. So the first thing, the first concept that we had in our mind is that the functions of proteins usually reside in the flexible regions of the protein molecule. And so which means that we have to look at it locally. And also, because we want to annotate novel proteins, our method has a non-evolutionary basis. And the dynamics, because it can relate to all parts of the protein, it's also a holistic aspect. As you can see from the right-hand side, this figure here, when we do, try to see the time scales of different motions that are ongoing in the protein, the bond vibrations are at the picosecond level, the side chain rotations at nanosecond level, the loop motions are in the microsecond level. So because the loop motions are primarily, determine the functions, so we aim to do a economical microsecond simulation. And in those days, doing microsecond simulations were still hard, not so much so with the advent of high-powered GPUs. But to fully conquer the collective motions, we should be able to go to millisecond level. So when we started this work, we faced several bottlenecks. Okay, so the time scale of motions that we were trying to deal with were microsecond and above. The time taken for each simulation was large, and because the computing capacity was limited in those days, GPUs were not that fast or are not so much available as well. The cost was also very high. And if you were doing a large scale simulation of 1 microsecond, the amount of the data that you acquired was difficult to store. I mean, those were the days where you did not have 4 terabyte disks, in a commodity scale. So we had a solution that we tried, which was coarse graining the whole thing. So what do you mean by coarse graining? When you do coarse graining, essentially it means that you replace uh, the protein molecule, each residue with some pseudoatom. And based on this pseudoatoms, you can represent your protein uh, using those pseudoatoms. In this slide, I'm showing you that this, through the cartoon, that you had all atom model on the left-hand side, and you just use the C alpha atoms, so you will get a representation just like a chain of beads uh, connected together in space. So to simulate such systems through pseudoatoms, you need a specialized force field, and you would not be able to use the normal uh, charm or amber or an OPLS force field. So in those days, we did not have many force fields. So we tried to develop our own force field, which is called the coarse-grained molecular mechanics force field. And Martini was available in those days. So we benchmarked our work against Martini. So the works essentially led to building of potential functions, which more than 1,000 in number. I cannot go into the great detail of these potential functions that we derived, but they were made in such a fashion, they were crafted in such a fashion so that they matched the NMR ensemble structures. So we validated, we took the NMR structures from the PDB, and we ran molecular dynamic simulations using the CGMM force field. And from the trajectories, we deciphered the fluctuation graphs. We tried to match these fluctuations graphs with the NMR ensemble data. And so this is one example that we have on the right-hand side for ubiquitin, where you can see the NMR structure is given by the dashed line. We have the CGMM, our force field, which is closely matching with the NMR RMSF root mean square fluctuation data. The Martini, which is fluctuating more than what it should. So we did these kinds of analysis to ensure that the force field that we derived, uh, faithfully represented the physiological fluctuation that we would be able to expect from the protein in the solution. And we have now this, the current state of this force field is such that we are able to do protein simulations for monomers and multimers, but we still do not have functions to incorporate ligands in it. So those are the new areas that we would be still working on in future. So once we finished, developing our force field, then we started, trying to figure out how we could go about using the trajectory that we got from our coarse-grained simulation and decipher if we can make a direct match between dynamics and function. So the first test that we did used a diverse data set of orthologous acyl-CoA binding proteins. And for these proteins, we also had the experimental binding and affinity information. And as you can see, the set of proteins that we took was from 21% to 82% sequence identity, which means there were sequences which also had a very poor match. And for those sequences for which there were no structural models, we built them through templates. Now, an advantage of using coarse-grained dynamic simulation is that you don't need to do any sidechain modeling for the protein. So it becomes very easy. So if you have the template, it's just replacing the amino acid names and just keeping the C alpha atoms and then joining them. So if you can look at this next graph, so after we did the simulation for 1 microsecond for each of the proteins in the test set, uh, the mm, the box here is the place where the binding uh, of the acyl CoA happens. And as you can see, the chain length of the acyl CoA is given here. So the bovine, the yeast, the M. perniciosa, and the rat has a medium long side chain. And the falciparum and the T. brucei has short chains. So if you look at the graphs here, the shorter ones are highly flexible, whereas the ones which are medium long, they are clustered together here. So this was the first test that gave us hope that whatever we were doing using the RMSF was useful. So the goal was to see, if we could identify all the flexible regions of the proteins and then use the flexible regions to actually match and see, which flexible regions of a given protein match with each other, and if they would, give us some kind of a signature. So the task of matching, first we need to demarcate flexible regions. So what we did, we discretized the the numerical values of the root mean square fluctuations for each residue that we got, and then we converted them into a string. So what happened is that when we converted all our RMSF graphs for each protein into strings, we could quickly do what is called a local search, a string-based local search, to identify all such substrings which we are matching. And we then empirically, figured out that if we had a criteria where the symbol L was greater than 35% or a combination of G, H, or I was greater than 14%, we could annotate these regions as flexible. So once we have identified all the flexible regions of the molecules, then the goal was to see if, if we take a library of proteins and for each protein we know all the flexible regions, now which flexible region would match with each other. Now we used something called an autocorrelation vector. We could have used the traditional way of eigenvalues to also do this match, but we prefer to use an autocorrelation vector because this is a concept that we borrowed from cheminformatics, where it is highly used and it is very simple to compute and also very fast. So if we just forget about all the formulae that are written above and just focus on these points that are shown here. So assume that you have a small segment and these are the C alpha atoms in three-dimensional space. So what you need to do is essentially you just find the distances between all the pairs, and then you decide what will be the span or the bin for your vector. So for example, 0 to 2 angstrom, 2 to 4 angstrom, 4 to 6 angstrom, 6 to 8 angstrom. So simply you divide the space into certain bins and you count the number of cases that fall into these bins. So then this becomes your autocorrelation vector. It's a very simple computation and very fast to do. So what we did, we computed the 3D autocorrelation vector, the unweighted vector for each of the flexible regions. And then we did a matching based on that. So here is one example here. So you can see this protein here for the whole 1 microsecond trajectory. We have discretized and and extracted frames for every 100 nanoseconds, which means that from the starting 0 and to the 1 microsecond, we have 11 frames. So you have 11 such vectors computed here. And similarly, for the next protein here, you have such vectors here, and you have such vectors here. So now you simply take these vectors and compute a simple correlation coefficient to get a matrix with 121 elements in it. And you can see that if two proteins have a good match in a certain segment, then you will get a very high match. And if they don't, you will get a very poor match. So in this case, this protein has a match with this protein. So you get a correlation matrix with many elements having a very high correlation, and which is not true for the other. So what we did, we found out the thresholds. So this was the criteria that we had. So if you had 25% of the elements with correlation value greater than 0.95, and at least 50% of all the correlation values greater than 0.9, then it would deem that the two proteins have the same dynamics. Or if the total number of cases greater than 25% is counted as Y, and these are counted as X, you could also calculate what's called an Euclidean distance to get a value. And using both of these, one could decide what is matching and what is not matching. So initially we took 60 arbitrary proteins, including moonlighting proteins, and we figured out that all of these have also experimental data available. So we took these, and what was encouraging for us was that when we found out the true positive rate, it was 87%, and the true negative rate was 92%. So with this, we kind of established a framework where we knew that we could work more with this method. The interesting aspect of the work that we did was that it allowed us to identify functions of proteins that are matching, where you would never expect to get any insight if you did a sequence-based search, or even a structure-based search. For example, you can see on the right-hand side, we have a protein where both are potassium channel inhibitors, and the global match is 9% only. And only in one section of the structure where the autocorrelation vector matches, you can see that this is the most important region due to which this potassium channel inhibitor is working. So you can see that the secondary structure is helical here, and this is kind of a loop that is there. But apart from this match, there's nothing else that will suggest that these two are potassium channel inhibitor. Similarly, we got matches where the secondary structures were different, and this is binding the vitamin pantothine for dihydrogen phosphate. So in this case also, it would be very difficult, just if you did a motif-based search also, to figure out that there's a match here, because the motifs won't match as well. But obviously, when you do a dynamics, the autocorrelation vectors match. Now, for finding the moonlighting functions. So for finding the moonlighting functions, you can see an example here is for 1WZT. In this case, the ancestral function is the biotin ligase, and the recruited function, the moonlighting function, is the DNA binding. And if we did a match, it was known, though both of these functions were actually known experimentally. But when we did this dynamics based search, we could also see that it could match to 2EJ9 for the ancestral function, and 2L0 key for the L0K for the DNA binding. So this way you can, find both the ancestral function and the moonlighting functions. And if there are other functions as well, you would be able to find them. So we try to, leverage these, analysis in our work that we did subsequently. And I will try to walk you through some of them quickly. So the first work that we did was to see if it helps us improve function annotation. So this is a schema. So what we did, we wanted to see that if there are proteins with cis peptides for which we know the functions, if we could leverage this there. Now cis peptides, as you may know, are a very conserved feature in the protein, and because cis peptides are functional. So we did an experiment where we just took the cis peptide segment and just used a sequence based search. And then we took just the dynamics of the proteins and then identified the autocorrelation vectors for the cis peptide segments. And we independently, we did the function annotation. And then we saw that what happened, and then what happened if we combined it. So when we did that, so this is the summary of the results. I'm just, for the sake of time, I'm skipping all the other details. So we had 204 such cases for which the experimental data was also known. And the detailed functional analysis was available in various published papers. So when we did the sequence only analysis, the true positive rate was 0.72. When we did dynamics, it was 0.88. And in this case, we did not use the Euclidean distance, but just the thresholds that we determined. But what you would see that when we do the whole accuracy, so ACC stands for accuracy. So when the overall accuracy we did for sequence based approach, it's 0.78. This is marginal, improved to 0.80, but when we combine it, then it went to 0.88. When we use the Euclidean distance based criteria, so this is a little bit more stringent. So we find that, although the number of cases have come down from 154 to 138, we have a more accurate, answers. So which clearly told us that if we use both kind of information, if we mix them together, then the chances of, improving our analysis is quite significant, because 0.78, 0.79, and 0.92 is quite a significant improvement. Another application that we did was through a blind test that we participated in last year, which is called the critical assessment of genome interpretation. It's called Kaji. This is, an Olympiad similar to Capri and CASP, many of which some of you may have participated. So in this Olympiad, you are given proteins and their mutants, and then you are asked to tell, which of these mutants are, altered. So there's an altered phenotype. So the question is, just the genetic mutation lead to loss of a certain kind of function? Okay, so that's the whole point. There were many other targets. So I'm going to, for the sake of time, I'm going to only tell you about two. So the first is a challenge in which you have to predict the alteration of the phenotype for 34 single mutants. Okay. Now this is a protein called CHEK2, which is a kinase protein. It's a very large protein of 586 amino acids. for which patient data was available for 34 persons, and one had to tell, whether these which of these mutant patients had a cancer or did not have a cancer. So basically, from these patients, these genome sequencing was done, and these mutation information was accessed. Now, CHEK2 is a very important protein because it is known to maintain genome integrity and also regulates the G2M cell cycle checkpoint. And also, it is required for the activation of the DNA damage repair and apoptosis of cell if there's a DNA double strand break. So any malfunction here leads to tumor and subsequently cancer. So this was known, and because this is a very important protein, so this was given as one of the targets. The second target that was given to us was for calmodulin. This is a cell survival protein. And for this, again, the data was available from 1,719 patients. So we had data for from so many patients. This protein is also known for cellular functions such as signaling, division, differentiation, apoptosis. And it has been found that the variants, there are certain variants of CAM that alter, they alter such that, they are present in heart attacks, cardiac arrhythmias. So we were asked, we were tasked to, predict the phenotype change in these two proteins. So this was our pipeline. So we did molecular dynamic simulation wherever model building was necessary. We filled in the gaps, and then we identified the flexible segments, and then the correlation coefficient was obtained through the autocorrelation vector, and we calculate the probability score or the similarity score for the CHEK2 and the CAM1 respectively. So the probability score is essentially the correlation coefficient, and the similarity score essentially identify the number of flexible regions in the mutant that were functional to the total number of flexible regions in the wild type. So this was the result that we got. So this is experimental results that was shared after the challenge got over. And you can see on the left hand side, these are cancerous and these are benign. So, and this is the probability score that we predicted, which means that these cases we predicted correctly, and these cases we predicted correctly. Then this is wrong prediction, and these are wrong predictions. So that is kind of summarized through some data here, where we have calculated the Pearson correlation, and we have staggered the fractions. So you can see that for about 2/3 of the cases, we are doing fine, which means there's not much difference between the predicted, experimentally predicted scores and the theoretical score. So, and if we relax the criteria, then we would say that, about 82% cases, we have some fair bit of results, but 18% cases, our results are anti-correlating, which means you can see the correlation drops from 0.47 to 0.02. So these mutations, we just, opposite predictions were made. So we have also done ROC plots, and you can see that we do have a positive signal for 75% cases. So for 75% cases, this is the graph here, which is a reasonable prediction. What is interesting is that when we compare our results with polyphen, so polyphen is one of the cases where you have machine learning involved. So we don't do that bad. So F1 is a classifier based approach. So we do 0.7 and they do 0.76. The accuracy is 0.6 and 0.7. So we do similar analysis for the CAM1, and I will not go into the detail of this for the sake of time, and I'll summarize the overall result. So in this case, as you can see, the accuracy for us was 0.73, whereas for polyphen, it was only 0.49. So you can see in this case, we have done much, much better than polyfen, which is a machine learning based methods. And even our classification score is much better. Now, one can ask that why we are doing much better here, despite very, a simple way of analyzing. So these are some of the things that one needs to go back and look deeply, where the functional parts are of the protein, and which helps us connect to the phenotype. So that was two examples. That were two examples that we presented for the Kaji challenge. The last one I will present is for a metal binding site prediction. As you can see, when you have a metal binding site that is absent in a protein, and you would want the metal binding site first to be deciphered before you can annotate function. So such kinds of methods that we have developed becomes very handy. So here are two examples. This feature plus is for calcium binding, and FoldX is commonly used for predicting calcium, copper, and zinc binding sites. So we have done built a method for predicting for these 5 binding sites. So currently, we in the PDB, about 62% cases are metal free and 38% are metal bound. But many of these metal free cases could actually have metal binding sites there, which, if annotated, would help in finding true function. So this was our motivation. And this is just, this is just pictorial to suggest that when we have dynamics information, then we know which ligand will fit exactly to which pocket, because when you do dynamics, the pocket shape changes, and such that which pocket will fit which one, which otherwise you may not have unless the static structure is exactly matching with the ligand binding pocket. So in our case, we have have a flow chart. In the flow chart, there are two sides. One is where you prepare the data base with a template library, and in our database we have these many structures which are incorporated. The total is given here, and this is the test set here. So what we do is we first identify the metal binding sites, and then use 4.5 angstrom radius, identify the residues, and then we have a question there that whether this is specific binding site or not. A specific binding site is one that is functional. So we have used OrthoDB database to find out which sites are specific and are functional sites. We take those and we create a template library using geometric hashing. So once we have done this, then essentially we have to take the query protein when we do the dynamics, and then we take the sample frames, and then we use the autocorrelation vector and we do the matching. So once we have done the match, then we enforce some ligand binding specificity on the matches, and that helps us to get the final prediction. So this is a paper that is currently under peer review. So this is a quick overview of what we do. So you have like 4 pseudo atoms, and in the center you have the metal atom. So you essentially store information for these 4 pseudo atoms, and then you do this for all data in your training set, and when you get the query, you build these things again, and then you match. So that's what you do. So this is schematic for that. So you have initial configuration, and then you do the dynamics, and then you will get all kinds of residues, which will be extracted from these conformations. And once they are done, then you enforce the ligand binding frequency. So this ligand binding frequency is pre-calculated based on the known propensities for metal binding, and a score is calculated. So this is the comparative result of what we have done so far, and the results for the test. So for this zinc, you can see the comparison is being made for two methods called MIB and IronCom. For those of you who are working in the area, you may be knowing about these two methods. So you can see that for the data set that is chosen for test, so we do 79 and MIB does 62 and this is 75. We do very good at 95 for iron and they do 81 and 74. For copper, of course, MIB is doing better than us, and IronCom is 77. For magnesium, IronCom is better. So we can say that for copper and magnesium, we are not doing as good as superior, not much superior than the others. But for copper and iron and zinc, we seem to have a fair bit of advantage in the method that we are using. So my time is up. So I will summarize what I spoke about today, the take-home lessons. So what we saw is that if we do simple analysis of protein flexibility to MD simulations, and we pass it through certain filters like autocorrelation vector, or you might want to use eigenvalues, eigenvectors also, that will allow you to match subsegments based on flexibility, and that'll help you to understand function in a much more robust manner, and which can be extended to understand phenotype and then diseases. So you can mix and match, you can incorporate this in your pipeline to improve function annotation and metal banding site predictions. This is a great way to understand moonlighting functions of proteins, and not the least, coarse graining has helped us to do these things economically, because if we were to use all atom dynamics, then I'm not sure if we would have been able to run so many thousands of MD simulations. So totally, based on the early work that we did on coarse graining, we were able to pursue the next set of analysis that I shared with you today. Now to acknowledge the people. So these work was done by 3 ladies in my lab. Pratiti, who started this work, she has already graduated, now a postdoc in Germany. Sritama also has graduated. So she works in a German company right now. Aditi has also graduated. She is working in GE right now. So these 3 ladies did this fantastic work in my lab. And if you want to use this, use the force field to do your own simulations and also try it out. This is available in my website. You can go there. So this is a snapshot of the web page, CGMM. So feel free to download it as a tutorial that allows you to go through the whole thing. And if you have any problem, you can contact me. So with this, I will end. My time is up. I'm sorry I overshot by 3 minutes. I'll be happy to take questions. Thank you.